Hello, my name is Guadalupe Bolaños, and my presentation is based on Herceptin. Now, just a little bit of background information. Herceptin is a monoclonal antibody that targets cell surface proteins. It was actually one of the first humanized antibodies approved for cancer patients. And humanized antibody just means that it originally comes from mice, but the genes encoding the antibody protein subunits were cloned, and the portion that encodes the antigen binding region was combined with human DNA sequences that encode the rest of the human antibody protein. And this was done so you would prevent an immune response in patients injected with the monoclonal antibodies that were coming from the mice. And so how Herceptin works, here we have a figure where Herceptin is binding to the HER2 extracellular domain, which is just a growth factor receptor produced by the ERBB2 gene, known as HER2. And Herceptin either targets intracellular or extracellular domain of the receptor, and it inhibits its ability to stimulate cell proliferation by either blocking the downstream signaling pathways or flagging the cell for destruction by the immune system, which of course will stop or slow down tumor growth. And of course, HER2s are overexpressed in breast cancer patients. So this is something that Herceptin is used to block. And next, I will explain the pathway that Herceptin interferes in a bit more detail. Here's just a simple diagram of what Herceptin does. Of course, as I mentioned, it's an antibody, and it blocks the receptor's ability to receive that growth signal. And with no signaling, of course, the cancer cell will either die or it will stop growing. And that diagram is shown in the right side. And now this figure here is just a schematic diagram of HER2 signaling pathways. So of course you have your ligand binding to the EGFR receptor, which will then combine with the HER2 receptor, inducing them. And the homodimers or heterodimers stimulate a series of signaling cascades, which activates your phosphatidylinositol 3 kinase and mitogen activated protein kinase pathways, which are the major pathways in cell proliferation. And normally, these cell signaling cascades will be terminated primarily relying on endocytosis of the complexes formed by EGFR receptors and their corresponding ligands. And then the EGFR receptors are either recycled to the cell surface or degraded by various enzymes. And based on these processes, there will be cell division, survival of the cell, proliferation, or any other will be maintained. And of course, under normal conditions, there will be normal cell growth and no tumor will grow. But of course, in breast cancer patients, with the overexpressions of your HER2s, that doesn't occur. So that's why trastuzumab, which is the other name of Herceptin, is used to inhibit that path, both the MAPK and PI3K pathways, which cause an increase in cell arrest and suppression of cell growth and proliferation by interfering the dimerization of the HER2 receptors and preventing it from activating and phosphorylating. And some of the side effects that are caused by Herceptin, some of the common ones, you just get your headaches, nausea, fever, infection, coughs, rashes, insomnia, or diarrhea. But some of the serious side effects, you get heart problems. It either reduces the function of your heart, you get congestive heart failure, arrhythmia, cardiomyopathy, cardiac death. Patients can experience swelling of legs or ankles, a weight gain of 5 pounds or more in less than 24 hours, or shortness of breath. And if they do experience this, they must seek medical help. Other serious problems are in your lungs. It could cause inflammations of the lungs, which is life-threatening, of course. They can suffer from pulmonary infiltrates, hypoxia, pulmonary fibrosis, and pulmonary toxicity. And patients can experience trouble breathing or just tiredness and fluid in the lungs. So my question for you is, what is one downfall, if any, of using a humanized monoclonal antibody as opposed to a mouse monoclonal antibody? Thank you.